in our department. Ted got his PhD at the University of Tennessee, working with Dan Simberloff and Jim Drake. He then did a postdoc at Landcare in New Zealand, working with Dave Wardle and Rob Allen. Taught for 3 years at the University of Hawaii, Manoa, before coming to Stanford in 2008. And I've been keeping up a bit on his work as we've been going along, but I'm really looking forward to having a good, thorough summary of it today. And please join me in welcoming Tad Fukami.
Okay. Well, thank you, Peter, for the introduction. And thank you, everyone, for coming to my talk today. Is this a good volume for everyone? Okay. All right. So I'm very excited for this opportunity to share with you today some of the work that we've been doing over the past 3 years or so since I moved here to Stanford in fall 2008. So I'd like to begin by acknowledging some of the current and former members of the lab because I'm going to mainly talk about these papers that we've recently published, including Melinda Belisle, who is a PhD student in the lab, and Mark Knopp, who is another PhD student who just defended his thesis last month, and Mifi Nakajima, who is a research associate in the biology department here, and Kabir Pei, who is a former postdoc in the lab, and he's now currently at the University of Minnesota. I also like to thank other members of the lab, including Ben Callahan, Mary Gauthier, Holly Miller, and Rachel Vanette, and as well as who we call extended members of our lab, who come to our lab meetings actually really regularly, weekly, and they always provide nice feedback on our work. And I also, of course, like to acknowledge the support, financial support that we are receiving from National Science Foundation's Thurman Fellowship and New Zealand Landcare Research. So I think nobody in this room would disagree with me if I said that one of the most important questions in biology today is about biodiversity, the diversity of life, where it came from through evolution, how it is maintained now or destroyed, and where it's going in the future. And there are so many ecological concepts that try to explain biodiversity and variation in biodiversity. But if you ask an ecologist, one of the most influential and important concepts, I think many people would say that theory of island biogeography is one of them, proposed by MacArthur and Wilson, as many of you know. Here, what they say is that you can understand biodiversity, particularly species diversity, in any island or any island-like habitat, for that matter, by thinking about just two things. One is how frequently species are coming from the mainland or the regional species pool to colonize these islands. And the other thing is the rate of extinction, how quickly species are going extinct. So species diversity is basically a net balance between the rate of immigration and rate of extinction. So there's a lot of emphasis on this concept, but actually this kind of island thinking with immigration as an important process affecting species diversity goes back much earlier than that. So one classic idea is that if you think about these different island community or any kind of discrete habitat, as long as you have the same environmental conditions like physical and chemical conditions, even if you have some variation in terms of which species come first and second and so on, if you have this kind of variation, you can call it A, B, and C, D, and so on. It doesn't really matter because as long as you know what conditions you have and what species you have available, it's always a predictable assembly in such a way that eventually you always end up having the same set of species. And we can call it species sorting because what's happening here is the species sort themselves according to the conditions defined by local habitats to get into the same kind of species in the end. And really, this kind of idea can be think of as something that was most forcefully developed and formalized by one of the founders of what we now call community ecology, a plant ecologist, Frederick Clements, who thought that the plant succession is a very orderly process that will end up with this deterministic species composition. Of course, that's just one of the ideas. Another contrasting idea that we have now is that assembly, community assembly, can be historically contingent in such a way that even if you have the identical conditions in local habitats and identical set of species in a regional species pool, just by having variation in species history, you can have a lot of variation in species composition among local habitats. And we call it priority effects because what's happening here is that the early arriving species are going to affect either positively or negatively the performance of late arriving species. And as a result, we end up having different species composition. And if you trace back to the concept, this kind of concept, you can think of Henry Gleason, another good botanist, as somebody who first think of this kind of ideas, most articulated this kind of idea. And now today, we always think of this idea as alternative stable states, meaning that eventually, at the end, when you have different species history, immigration history, you have multiple set of species composition that you can end up with, depending on what history you have. And once one community get to one state, unless they are heavily disturbed, they cannot move to another state. And this kind of alternative stable states idea has been really popularized first by Jared Diamond when he published the book chapter in 1975. And here, what we see here is island of Papua New Guinea. And he looked at a distribution of different bird species, and he called it checkerboard pattern, distribution pattern, where closely related species can be found, only one of them can be always found in a given island. And he thought that there is no other explanation for this kind of pattern other than the fact that maybe these species are competing, and this is just we are seeing as a result of alternative stable states. Of course, that study has been highly controversial, but after that, over the past 3 or 4 decades or so, there have been many, many more studies suggesting the potential importance of immigration history. So it's kind of expanding in concept because it's not just ecological phenomena, but now it's an evolutionary phenomena as well, because if you have the same species, sometimes that species give rise to new species, but even with that, the same species can sometimes just don't succeed in providing new species, depending on what history they have previous to colonizing these communities. And there are a lot of indirect evidence coming from, for example, lizard communities studied by Lossos, or plant in Micronesian island by Silvertown, and we also were able to provide experimental support using experimental population microbes for this kind of idea on effect of diversification. Another way that people have been expanding this concept is it's not just community structure like species diversity and composition, but it could also be community function that's also affected by history. So I'll just show you 3 examples here. For example, there is a study showing that the total community biomass of algal community can be affected by the history or the plant community biomass. And we also had a study looking at wood decay fungal communities and showing that decomposition and nutrient cycling can also be affected by history. So what we have is this possibility, this causal relationship, simple causal relationship, where history is affecting biodiversity. And I'll just replace biodiversity with more general term, community structure, to describe not just diversity, but also composition and other aspects of community structure. And this is what I'm going to call historical contingency. And as I just mentioned, it's not just structure, but function like decomposition in nutrients and so on. That's also affected by function, and function can in turn affect structure. So when you think about it a little bit about this kind of historical contingency, this is a scary picture to ecologists because we don't really have, usually in most cases, we don't have any information about immigration history for any given community. So if history is really important, we are kind of stuck in trying to understand structure and function of ecological communities. So you can see there's a great temptation in wanting to believe that Clemens was right, that the community ecology of predictive science, where things are all deterministic, but more and more, as we study more ecological communities, we are realizing now that community ecology may be a historical science. Maybe Gleason was right. So I think what we need to do is to think of a different kind of question to think that maybe specific community structure and function may not be understandable unless you have historical information. But you can kind of change the question a little bit. And this is the central question I'm going to think about today. When should history matter? Instead of trying to understand any specific community structure or function, we're going to ask if there is any way to have understanding of when we should expect to see history to be important and when we don't need to worry about important. And that's actually a question that's becoming more and more popular over the past 3 decades or so. And so what we do is to think about how, in what ecological context, history can become more important, and under what other ecological contexts history may not be important. Just to give you a few examples, I'm just going to use my own example here. I was able to show with the protist community some time ago that it's when we have a small habitat, that's when history becomes really important, whereas if you have a large habitat, that's when communities become more predictable. Another example I can show you, there is a really nice work done by John Chase, published 2010, showing that when you have a nutrient-rich habitat, that's when history becomes very important. And I'm not really telling you why that is, but this is just a quick overview. And here, if you have a nutrient-poor habitat, on the other hand, that's when you should expect to see more predictable community assembly. So I think there is nice, really active, ongoing work right now trying to improve this kind of framework right now. What I want to do today is not so much about ecological context, but I'm going to focus more on what I'm going to call evolutionary context, which hasn't been studied actually nearly as much compared to ecological context, but I'm going to argue today that what I'm going to call evolutionary context can be just as useful or important in understanding this question, when should history matter? So that's what first thing that I want to do. And second thing I want to do is to think about this idea of alternative stable state that's really popular. And now, instead of thinking about stable states, I'm going to argue, I think this is going to be a little bit controversial here, but instead of thinking about stable states and focus on that, I'm going to argue that it's very, very important now to think about transient states instead, and why that is critical for thinking about historical contingency. And I'll explain to you what I mean by this more as I go through this talk. So in my lab, there are a lot of people in the lab is working on many different systems, and we like to use many different systems and many different organisms because each question, specific question within this broad framework can be answered more effectively by different kind of systems. But I have only 45 minutes, so it's not possible to talk all of this in one talk. So what I'm going to do is to give you a flavor of the kind of answers that we are trying to give to this central question, when should history matter? So I decided to tell you about 3 things. One is that I'm going to say that one answer you can give to this question in terms of evolutionary context is that when history should matter more when immigrants are closely related to one another. And I'm going to use Nectar microbes at Jasper as an empirical example. A second answer that I want to talk about is when history should matter more when immigrants are poorly adapted to new ecological contexts. And I'm going to use bacterial experiments to give you evidence for that. And the third thing, as I said, is about transients, that I'm going to say that the answer to this question is going to depend on whether we look at stable and transient state. I'm going to use computer simulation work to talk about it. And I'm going to explain why it's important to make distinction between transient and stable states in thinking about historical contingency. So first, about Nectar microbes, which is fun at Jasper. So as I said, this is the ecological context that a lot of people have been focusing on right now. And as I say, I'm going to think more about evolutionary context, more specifically where the species came from in the first place through evolution and how that might affect historical contingency. And as you know, with a lot of molecular methods now, there's a lot of information about phylogenetic relatedness of any kind of organisms now. So we can use this kind of information now to think about how this kind of information may help to understand the importance of immigration history. So that's the question we wanted to ask. And this is the work that was done by Kabir and Melinda and me. And so we thought about this question, and basically what we are asking is if there's any relationship between phylogenetic relatedness between potential colonists and the importance of history, or in other words, strengths of priority effects. We thought about it, and I think there are many ways, many expectations that you can think of, but for simplicity, we thought about just the competitive interactions where species are competing for the same kind of resources, and based on that, we think that the simplest way possibility is just simply this, that if you have a more closely related species, then in general, that means that the species are more ecologically similar, so they may be requiring the same kind of resources, and also they may be competitively more similar to each other. So when which species come to a community may matter more when you have a closely related species as opposed to distantly related species. So that's the kind of really basic, primitive kind of expectation that we had, and we wanted to test that, but experimentally, using nectar inhabiting microorganisms. So here, this is a system that I started to work on after I moved here to Stanford. And what we have here is that when you look at flowers, you have nectar. And in nectar, there's actually multiple sources of yeast and bacteria you can find. And this is a good system, in addition to being an interesting system to learn about in and of itself. We thought this would be a good system to think about community assembly for several reasons. One is that, like many other microorganisms, microbial systems, they have short generation time and rapid growth. So if you are experimentalist, it's inconvenient to have this miniature ecosystem to do a lot of community assembly experiment. Unlike most other microbial systems, it's a simple system because we have only about a dozen species that can tolerate this kind of harsh condition. Nectar is a sweet thing, but it's actually a harsh condition because it's a lot of sugar content, and also there could be antimicrobiotic compounds in nectar. And because of that, we have a simple system with relatively few species. And also, when you think about their habitat, it's a well-defined island-like habitat because you can think of flowers as their local communities. And when you think about immigration, which is our focus of always the research, immigration happens via pollinators. In this case, if you know monkey flower, you know that it's pollinated by hummingbirds. So we have a lot of video clips now because we want to track where hummingbirds go. So here's an example here. This is taken by Jasper Ridge. So when you think about how hummingbirds are going to flower, what they do is obviously their motivation is to get nectar, but they probably don't know that. But if they get also yeast or bacteria, then when they go to a new flower, they can help those yeast to immigrate to a different flower. So this is the immigration that we are dealing with with this system. So Melinda did a lot of DNA sequencing to get species from the Jasper Ridge. And what we decided to do is to focus on these 6 species. These are the species that can actually actively use nectar as resources. And we thought of that as our species pool. And because we identify them anyway by molecular method, we have a good information on how they are related phylogenetically. And so we wanted to do an experiment, and we wanted to use real flower, but instead of doing that, to ensure that we have a good experimental control, we did this, used this, what we call artificial flowers. And here we put real nectar. This is sterilized and filtered, so there's nothing initially like a new flower we have, and 9 microliter is within the range that you see in real flowers. And as a simple experiment, we just picked one of the species and introduced them on day 0, and then we waited for 2 days to introduce another species, and we looked at how abundant they are by using molecular methods to see their relative abundance on day 5. And as another experimental treatment group, everything is identical except that now the interaction order is reversed. And we also wanted to have these control as single inoculation controls. I wanted to give you some natural context to this experimental design because I think it's important in trying to apply what we see here to what's happening in nature. First, we went to Jasper Ridge just to see how long these flowers last, and it turns out it's about a week. So actually, if you do an experiment for 5 days, that may not be so long for us humans, but for microbial communities, it's a long time because it's a majority of the time of these communities. Another thing we looked at is we just measured a total abundance of microbes in real flowers at Jasper Ridge, and it turns out that only a few days is enough to apparently for these communities to reach some kind of carrying capacity. So 5 days, once again, is a long time relative to the short generations and growth of these microbes. Another thing we looked at is it's a convenient flower because you can look at female reproductive part of these flowers to see whether they've been pollinated or not by looking at whether stigma is open or closed. And we actually went to Jasper Ridge and just measured a proportion of flowers that have stigma closed or open. And it turns out that it actually takes several days to see a gradual increase in the proportion of number of flowers with stigma closed, which indicates roughly, it's only roughly, but which indicates that if you think about immigration of different yeast, and that most yeast species rely on hummingbirds, as I said. So if they're coming so slowly, maybe this 2-day interval is not that unrealistic when you think about how frequently different species are immigrating to these small communities. And this was a lot of work done by Kabir, but we had all possible species pairs tested of those 6 species, and I'll show you results now. So what I'm going to show you now is I'm just going to focus first on green species and see how well they were doing. And we had these competing species as a second species. So this is the initial density, and this is how abundant they became after 5 days. As long as this green species was the first one to be introduced, there's no difference. And even the monoculture control is the same as the other treatments, meaning that they were not affected at all by a competing species. When they were the second species to be introduced, things are different. Here, for example, depending on which species you use as the first species, there's a complete suppression of these species in terms of abundance. This is going to be a busy slide, but I'm going to show you all the data because we got them. So, so here's everything that we have, and there's a lot of things going on here. So I want to highlight just a few things here. I want to start with this species pair, blue and brown, or I think it was orange, brown. So here, this is one of the strongest case of priority effects. You can't predict which species is going to be abundant unless you know which species was introduced first. So if you look at blue species, they do very well as long as they are the first one, but not at all when this was the first one. It's the mirror image here. As long as this was the first one, they're just as fine and not affected at all, but they're completely suppressed if they were the second one. So this is the extreme case of strong priority effects. Now I'm going to look at this species pair, and it's similar to the first one, but a little bit different in the sense that if you look at green species, they do fine as these ones, but they're not completely suppressed, and they're still hanging there even if they're the second species. So this is this kind of intermediate, and I'm going to show you the last example here. Here, if you look at this species pair, it's the other extreme where the history doesn't matter. Whenever history you have, it's always the blue species that wins. So looking at all these, it seems like there is good variation in terms of the strengths of priority effects. So what you can do is to use all of this information now to go back to this question about the relationship between these two. And because we have this phylogenetic relationship, we can plot that on x-axis and then quantify strengths of priority effects here. And as expected, you can ignore this outlier, but you can ask me later what I think about this. But generally speaking, as we expected, there is this significant relationship between relatedness and strengths of priority effects, where priority effects is stronger if you have a closely related species. And we're also trying to give ecological explanation to this pattern. And we found that by measuring growth in different sugar conditions and also their resource use, we're finding that there's actually also a relationship between phylogenetic relatedness and ecological similarity between different species. So I think we have more work to do because it's just the pairwise interactions and we are just looking at a subset of species here. But it seems like we do have evidence, and to our knowledge, this is the first experimental evidence showing that maybe it's when you have distantly related species in your species pool, that's when you should expect to see predictable assembly, as opposed to this case where you have a lot of closely related species in your species pool. That's when you should expect to see a lot of historical contingency in community assembly. As next steps, we are working on this system a lot now, and Rachel Vanette, the new postdoc in my lab, is a chemical ecologist coming from University of Michigan. And so we are looking at different amino acids, sugar, peroxide, and pH, and different kinds of chemical properties of nectar to provide mechanistic explanation for priority effects. Another thing that we're excited about is to think about not just community composition, species composition, but also community function, not just structure, but function. And when you think about function of flower, everyone knows that the function of flower is plant reproduction. And what we are doing now is experimentally add different microbial species to real flower, the nectar of real flower, and see how that might affect pollination. And we are actually getting promising results. So we looked at pollinator visit, or pollen deposition, seed production, and even seed germination. And all of these are showing that depending on which species you introduce, there is some effect. Plant reproduction is more successful when you have certain microbial species. So we want to link structure and function in this way. So that was about flower microbes at Jasper Ridge. And now I'd like to move on to the second answer that I wanted to give you, which is that history should matter more when immigrants are poorly adapted to new ecological context. And this is the work that we did with bacteria in the lab. So I'm going back to this picture, and I was with the nectar microbes. What we are doing was to focus on this evolutionary context as a relatively neglected factor, but we haven't really thought about interaction between evolutionary context and ecological context. When you think about evolutionary context, that's not separate from ecological context, because the adaptation, for example, of species in a species pool and what they do, these species, once they colonize this new habitat, depends obviously what kind of adaptation they previously had before immigration. So that's the kind of question we wanted to ask now. How previous adaptation history of species in a species pool to the kind of habitat conditions that we have in local conditions, how that may help us understand the importance of immigration history. And this is the work that Matt Knopp did with me and Samantha Ford, faculty at UC Santa Cruz. So like the first question, we thought of this relationship. And so now the question is, can we use adaptation to new ecological context to predict the strengths of priority effect? We couldn't think of any good prediction, so I'll be honest and say that this is left as question mark. And we just decided to do experimental test here using this bacteria species, Pseudomonas fluorescens. And this is a nice system, we thought, for asking this kind of questions, because this system is good for thinking about not just immigration and extinction over ecological time, but also evolutionary processes like diversification that happens in a new habitat. Because there's one classic paper in 1998 showing that if you put this species in the lab condition, and I think it's important for me to say here that this bacteria originally came from plant-associated strains. So this kind of lab conditions is really foreign to these bacteria. But anyway, there are people who found that if you introduce this ancestral genotype, this vial, they can quickly go through diversification and adaptive variation even within just 4 or 5 days to give rise to new mutants in these conditions. So what we wanted to do is to use, as a simplest case of manipulating history, we wanted to have just 2 genotypes. One is an ancestral genotype that was taken directly from nature, from originally plant-associated bacterial strains, and the other one that I'm going to call evolved genotype, and I'll tell you what I mean by evolved genotype in a minute. But just like Nectar microbe experiment, what we wanted to do is to do a simple kind of experimental design to manipulate historical differences here. So here we have two genotypes introduced sequentially as one of the experimental treatments we had, and we did another one, just like this, the previous one. This treatment is identical to this one, except the introduction history. And we also wanted to have controls to see what they would do if they were introduced alone. And so getting back to what I mean by evolved genotype, what we decided to do is to have two sets of experiments, the same thing, but only difference is that now the evolved genotype has either been poorly adapted or well adapted to lab conditions. And we did that by simply allowing the ancestral genotypes to grow in under lab conditions either for a short period of time or long period of time, corresponding to about 10 generations or 100 generations of bacteria. So because they are not well adapted to this kind of lab conditions, they are known to adapt to this kind of lab condition and evolve. And we wanted to use these poorly and well adapted strains to think about this question that we had about importance of historical contingency here. So what I'm going to show you the contrast between results for poorly adapted strains and well-adapted strains. And for each of these, I'm going to show you what happens if you use this kind of sequence of introduction and what happens if you have this kind of sequence of introduction. And here, so this ancestral genotype is introduced here, and then later, one day later, this was introduced then as a second immigrant. And if you compare that to what happens here, there's a clear difference here in terms of number of morphotypes that emerged from these genotypes. So if you just focus on evolved genotypes for now, it's a clear case of history being important in terms of the extent of diversification that happens as a result of either first introduction or second introduction. You can look at the same thing for well-adapted strains, and we did the same thing, as I said, with the only difference being that this genotype is well-adapted now. Somewhat similar to this, they don't do well in terms of the number of morphotypes they give rise to if they are the second strains to be introduced. If you introduce this strain on as a first immigrant now, somewhat different from this. Now, if you just focus on evolved genotype, and there's no frog in this bio, a duck. So that it's easier to see, I can just shift it to make it more directly comparable. So big difference here, history being important, not important here if you have a well-adapted strain. So there's something going on that changes the importance of history. This is just another way of summarizing this result. So if you look at poorly adapted strain, then they are severely suppressed, so we have a negative priority effect happening, so this corresponds to this. And if you have well-adapted strains, there's no difference, so history is not important. We thought about what's happening, and although we don't have a direct evidence to narrow down exact mechanism, I can tell you that there are different papers that we can use on this system to give you actually very likely explanation of what may be happening. And you can think of fit landscape metaphor to think about what may be happening. When you think about poorly adapted conditions, so here we are thinking of plant-associated bacteria that's just thrown into this totally novel environment under lab conditions. And if you just let them adapt just for a short time, this is what you would see as poorly adaptive genotype. And so in terms of their potential to diversify, they do have still a lot of potential to diversify because they can search these different adaptive peaks. But as they adapt more and more to climb this mountain, what happens is that now this genotype is well adapted to this one particular adaptive peak. And once you have that, because this is now very competitive, even if they give rise to new mutants, they are competitive suppressed, so there's no mutants that can coexist with the original one. And you can think of also different kind of genotypes, and this is also becoming difficult because this which requires a mutation of a large effect, and also if that's not the case, they have to go down the adaptive valley and climb up, which we know from population genetic work to be difficult. So going back to this, I think what's happening here is basically the well-adapted genotype just cannot diversify regardless of the immigration history, which result in insensitive diversification in terms of history. And here, if you have a poorly adapted genotypes, they can diversify if they are the first one to be introduced, but they're completely suppressed if they're the second one. That's why we have this difference between poorly adapted and well-adapted genotypes. So going back to this central question, when should history matter? I think this is just one bacterial microism study, so I think it's important to be careful and not to extrapolate too much. But having said that, I think general idea may be broadly relevant here, so that when you have a species in your species pool that are well adapted to these local habitat conditions, maybe that's when you don't need to worry so much about historical effect. And when you have a poorly adapted species in your species pool relative to these local conditions, maybe that's when you have to think about, you need to expect a lot of variation due to immigration history. As next step, one of the things that I'm doing in the near future is to collaborate with some of the colleagues that I have in New Zealand and to try to actually apply this kind of idea that we got from bacterial experiment to other systems, maybe more larger scale systems. So in New Zealand, there's a lot of really good high quality data on phylogenetics and also abundance across the country. And so we're going to use this kind of data to think about how we can use phylogenetic information to see whether we can test the idea that we got from this bacterial experiment. And this, by the way, is a paper that was just published last month. They used this data. And so our plan is to expand this kind of work in the future. As another direction that I wanted to briefly tell you about is another postdoc, Ben Callahan, who is supervised with Daniel Fisher in the Department of Applied Physics and me, so he's co-supervised. He's doing a lot of more experiments on the bacteria, and his purpose is to think not just about resource competition, which has been our primary focus, thinking about competitor interaction, but he's also going to look directly at what he calls niche construction, which is another kind of process that can lead to priority effects. And think about these two, both of these two, to ask questions about priority effects. And if you're interested in this, I don't have time to tell you more about it, but he has a talk tomorrow at 1:30 as a part of the symposium that Daniel Fisher is organizing. So you can go to his talk if you're interested. So that was the second answer that I wanted to talk about. Now, as a third part of my talk, I want to think about transient state with you. And this is a work that we have been doing more as a theoretical work rather than an empirical work to start with. And so, going back to this picture, as I said near the beginning of my talk, there's a lot of emphasis on alternative stable states as the central conceptual framework that a lot of people are using now to think about trying to understand historical contingency. I think it's a great kind of framework that we can use to think about historical contingency, but I'm going to destroy it. So here the focus, my framework is that we're going to focus on alternative stable states to think about importance of immigration history. That's always what people do, at least when we have a theoretical work. And it's convenient because it makes questions mathematically tractable. But as we all know, as ecologists, that the community assembly takes time. And so we do have this kind of transient states. Once the community is destroyed by some disturbance, they have to start a new round of community assembly. And it can take a long time, actually, to get to the final state. That doesn't necessarily mean that it's a bad thing to focus on alternate stable states, because sometimes by thinking about where they eventually would go, even if they actually don't go there, you can use that information, that kind of prediction, to think about transient state, because transient state may be more or less comparable to what you would expect at a final point. But I think that's an open question, and I wanted to say that we need to think more carefully and explicitly about this question. Should we really directly study transient state, or is it okay to just keep focusing on stable state to understand historical contingency? So that's the question that I've been working on with Miffy Nakajima, and I have also another collaborative project with her at home, raising a new baby. But I'm going to talk about computer simulation work now. So what Miffy and I decided to do is a theoretical assessment of this idea about alternative stable states. And we did, she's a good theoretical population biologist, so we decided to do a simulation of plant community assembly. And what we did is a numerical simulation, and on a computer, we had 10 identical habitats, identical in the sense that they have a local same habitat conditions. And we had 30 species that vary in ecological traits, and in computer, we assembled initially empty habitat by sequential introduction of different species. And so important point here is that all of these 10 identical habitats that we set up in a computer are identical, except the fact that they are randomly assembled with sequential species interaction. So whatever variation we see in species composition here can be attributed to the random immigration history that were different between different habitats. And what we did, or are doing, is to think of different kinds of ecological contexts, like how isolated these habitats are relative to this mainland, or how heterogeneous habitats are, or how harsh these habitats are in terms of mortality rate of different plants. And we are finding a lot of interesting results, but I just focus on habitat isolation to just to make the main point that I wanted to give you. So after a lot of computer simulation, after 1,600 years of plant community assembly, this is when we made certain that all these communities have reached stable states. And I'm plotting how habitat isolation is related to variability in species composition among these habitats. And as you remember, you can interpret variability in species composition as indicating the importance of history, because they are different only in terms of random history. So this y-axis, I can actually label it just the degree of importance of priority effect or importance of history. As you can see here, habitat isolation doesn't affect the importance of historical contingency so much. Maybe a slight increase if the habitat is not so isolated. These are more variable. But overall, there's a lot of variation and there's not much differences. So if you just focus on a stable state, you would say that history is not that important, and the importance of history doesn't depend so much on habitat isolation. That's what you conclude if you just focus on stable states. Now I'm going to show you the same graphs looking at all transient states. Here, it's different. As you can see here, if you have a more isolated habitat, then history is more important compared to less isolated habitat. And that's the same from 20 years, 40 years, 80 years after we initiated communities. So there are two key questions you can ask. One is, are transient and stable states comparable? If they are, then it's okay to just focus on stable states, because what you see in stable states are going to be the same for transient states. And transient states are usually what we actually have in real communities out in the field. It's obvious that the answer is no. This relationship is different from these two. The second key question is that, are transient states short-lived? And this is all relative question, and I want to give you some bit of background here. So is 20 years long, 40 years long, 80 years long? And it's actually a long time, because in this computer simulation, each population of species, a plant species, need only a few years. Once they get to the local habitat, they need only a few years, 2 or 3 or 4 years, to reach their carrying capacity. So relative to 3 or 4 years, 20, 40, 80 years is a long time. In addition to that, in our simulation, by 80 years, on average, each species will have, on average, four chances to immigrate to any given local habitat. So they have multiple chances to get to different habitats. So 80 years, we are not really seeing initial trivial state where not all species haven't had a chance to immigrate. So for that question, I think we should say that definitely no. Transient state can last long time, and they're different in terms of this shape of relationship compared to stable states. So I want to briefly tell you why we have this kind of increase in variability in species composition as a function of habitat isolation in transient states. And the mechanism for that is interesting, which is something about plant-soil feedback. One thing I didn't tell you, but actually interesting, in our model, we deliberately modeled what we call plant-soil feedback, where you have plant species A that's affecting condition of soil such that as a result of growth of plant species, now you have a different maybe soil food webs. And there are two possibilities in terms of what people call plant-soil feedback. Here, you can think of maybe pathogenic fungi or bacteria. Because of the growth of this plant, subsequently, once this plant individual died, this soil is not good for the individual of the conspecific individual. So this is the negative plant-soil feedback. You can also think of a positive plant-soil feedback where this plant is going to condition this soil community such a way that this soil becomes better. So this individual later time is going to grow better. So this is positive plant-soil feedback. And there is actually a lot of momentum now with a lot of people working on this plant-soil feedback. But the point I wanted to make here relative to our computer simulation work is that because of this complex plant-soil feedback that we built into our model, what's happening here is that if you have an isolated habitat, that's when you have a lot of turnover of species, because one species is going to help some other species but not others. And once that species comes, then that species is going to help or harm some other set of species. So there's a lot of species turnover going on if you have isolated habitats. And that doesn't happen if you have a less isolated habitat. So that's why we have this kind of increase of importance of history. More plant-soil feedback and causing turnover of species being more important if you have isolated habitats and not so much in less isolated habitats. So this was just a little bit of what we are doing with computer simulation, but I think it's enough to tentatively at least conclude that to the answer to this question, should we study transient state? Yes. We need to study transient state explicitly, and we can't just focus on stable states to think about historical contingency. Because, for example, if you look at, going back to this picture, if you look at stable states, maybe you say that regardless of habitat isolation, broadly speaking, it's always a predictable assembly. But that's not the case if you directly look at transient state. When you look at a transient state, if you have an isolated habitat, you say that it's very, very much historically contingent if you have an isolated habitat as opposed to predictable assembly if you have a less isolated habitat. I don't play golf, but this metaphor makes sense to me, so I hope this makes sense to you too. If you play golf, it's not where you start and where you end that's interesting. It's where you go in the trajectories of where you go. That's interesting and important. And in the same way, if you want to understand ecological communities, you have to think about transient trajectories to really understand historical contingency. And so as next step in this part of work that we are doing, we're excited to work more and more with empiricists who is getting a lot of good data on plant-soil feedback, because I think it's a promising good mechanism for priority effects and not much is known about it. So I'm organizing a symposium at Ecological Society of America meeting in August to get together with a lot of good people working on plant-soil feedback so that we can give more empirical basis for plant-soil feedback in our computer simulation work. So I talked about all these three answers. And by putting it all together, the one take-home message that I can give you to summarize all of this is that to understand when history matters in community assembly, we need to look at the evolutionary background of immigrating species and transient dynamics of assembly in communities, in addition to a lot of work, in addition to ecological context that's been worked out a lot nowadays. So going back to this question, is community ecology a predictive science or historical science? I would say that this question can be reconciled because we should embrace historical contingency by studying historical contingency directly, but that doesn't mean that community ecology is a historical science, but because by changing the question, we can predict a different kind of prediction now. We can predict when history should matter and when not. And that's still a prediction that's useful. So I think we have a new generation of community ecology to merge these two people together and to have a long-term goal, raising community ecology as historical, a predictive science. And I think we have to raise this community ecology to make it this kind of historical yet predict. So that's the keyword that we're going to have in our research. And I think the way to do it is to pick different systems as we've been doing. And this is just the beginning. This is the end of my talk, but this is the beginning of our research in the lab. We're going to use different systems because they each have different advantages and disadvantages. So we are going to carefully select different systems to try to achieve this long-term goal most effectively. I think that's the end, and thank you very much, and I'm happy to take any questions.
migrating into a community as homogeneous genetically. That is, one member is the same genetically as the next member. But in cases where a small number of individuals are involved, like let's say inoculation of yeast into nectar of a plant, and of course genetic variation of the members of that species could be really important because it's only a few members that are, right? And then you have individuals and they're genetically different. How do you take into account genetic variation in the population in thinking about these questions?
That's a great question, and so far, as you can tell, we kind of ignore that aspects of finding immigrants, but it is important. So actually, we haven't directly looked at that question yet, but there are other people, particularly in Belgium, they study Daphnia communities, and they experimentally manipulate genetic variation of founding populations, and they do find, as you expect, that the genetic variation of individuals of the founding population can affect, obviously, what happens to community assembly. So that can be another, you can put the genetic variation as part of evolutionary context. I don't know, maybe it's here, but I think it's here, that genetic variation of founding populations as another factor that may be helpful in thinking about when history is important or not. That's a great question, and I think we might be able to look at that by looking at nectar yeast, because they do vary genetically. And I think that's one of the questions that Rachel was interested in asking, but there's so many questions that I don't know if Rachel is going to actually work on that.
So the transient states, the length of the transient states in the simulations, you concluded that it was mostly because of the feedback, the soil, basically. So a plant, the first species comes in and it has time to condition the soil more. If there's longer amounts of time for it to condition the soil, then that makes the transient states a lot longer. Is that?
Yeah, I didn't explain that too well, but in the simulation, it's just one. So we assume that once the plant comes, it only takes 1 year to change the soil conditions. So once they get there, immediately the soil changes, and then it will affect the other species. So that's not really the reason why we have this prolonged turnover of species. I think it's more have to do with the fact that we have this complex network of plant soil feedback that if you have a given plant species arriving, then to some species, it's good. But some other species in the species pool, it's not good. So if you have a slow immigration happening, they just have to go through a lot of turnover and tryout to get to the final stage, and it just takes the time, and that results in species turnover.
If you take out immigration and you just start with a fixed species pool, and then you ask how long does that take for it, like size to stop fluctuating, is that usually pretty short? There's no continual immigration.
It can be long. So you're asking if we put all species together at the same time, what happens to sorting of species? It depends on different things like how heterogeneous habitats are and so on. So it can take a long time. So I think we need to do more to be perfectly certain that it's the turnover actually that's causing the difference between isolation effect.
Sort of following up on that, have you looked at how the number of species that you have in that model affect like how long it takes to hit that stable state? Because if you have fewer niches that can be filled, maybe you will see as much productive persistence going on.
Yeah, that's a good question. So Sandeep's question was, if you have a different number of species in the species pool, how that's going to affect the importance of plant-soil feedback and community assembly. I think we haven't done that so much, but I would expect that if you have a fewer species in the species pool, that can make the transient stage shorter. So maybe that's another thing. How many species you have in your species pool, that's also going to affect historical contingency. Actually, that's one of my dissertation chapters. I didn't look at plant-soil feedback explicitly. I had a more even abstract computer summation as part of my chapter, and I did find that if you have a larger species pool, that's when you have more prolonged turnover, which result in the greater effect of history. Maybe start with you.
This is going back in the past a bit, but the way that people used to think about ways you could make assembly more predictable had to do with the properties of the species and their ability to colonize. So you're colonizing species with different properties in random orders, where another form of predictability is colonization is likely to be anything but random. Some things are really good colonizers, some things are really slow. Have you integrated those two strains of thinking about predictability at all?
Yes. So, of course, as Peter pointed out, in reality, the colonization or the immigration order is not random in many real communities, because some species are better at dispersing into different places and some species are not. So we did look at that question in our computer summation by assuming that there's a trade-off between competitive ability and dispersal ability, which some people think are, not all people, but some people think are a good assumption to have the trade-off between those abilities. And of course, it's just one way to stimulate communities, but to my surprise a little bit, if you include that kind of differential ability to immigrate, that doesn't really affect the picture we have here. But having said that, this is just one way to model communities. So I think there's much more to be done. And it's possible that if you assume that kind of trade-off, the history becomes less important because simply because the history becomes more predictable, as you point out. Liz?
So nice podcast. One question is probably isn't everything transient in some way? So in a sense, especially when you start thinking about the topic that Tim raised, the evolutionary dynamics of the species you're looking at, how do you decide that 20 years versus 1,600 years versus 16,000 years? Where is your stable state?
I think stable states can be defined only theoretically. And so I think your point is well taken, that it's maybe everything is transient in the real world. And I think that just makes transient states more important to study. I feel like I'm dodging your question. Maybe Alan?
Yeah, in your first two parts, you were talking about experiments, and then in the last part, you got into the theoretical calculation. In those first two parts, were you implying that the effects you were seeing were affecting stable, permanent states, or just that everything, that in addition to these factors that you saw of adaptiveness and such, you were always looking at a transient state?
I think we are looking at transient states, not, yes. And that's what empiricists do, of course, because communities are transient. But theorists would argue that we need to think about where they would go if they are given enough time. But I think there's limitation to that. Noah?
Yeah, so it struck me that some of the factors you discussed that promote an important role for predictability versus an important role for historical contingency are quite analogous to factors that promote an important role for genetic drift versus natural selection in a population genetic system, like the nutrient poor system being like a case where there's strong selection. Selection would be driving what lineages succeed rather than drift, whereas say, the small habitat is analogous to a case with a lot of drift. And I'm just wondering if there's any potential gain from thinking about some of the dynamical models in population genetics for the theoretical modeling component.
Yes, I think so. So slowly, community ecologists are just now realizing that we have a lot to learn from population geneticists. And there's a really good review paper last year by Mark Villand in Canada, and showing that community ecology can be understood by just borrowing the concept from population genetics. So we have drift, selection, and mutation, and so on. It's as you point out, it's really analogous to supply of new species and competition and so on. So I think there's a lot to learn from population genetics.
Well, we will all have plenty of time to talk with Tad. So thank you very much, Tad.
Thank you. Thank you. Thank you.
Nice job. Very easy to understand.
Oh, good.
It was really nice to see how the